Hello, everyone, and welcome back. We are now looking at the biomedical devices, innovations, and material solutions in the excretory system. And what we're looking at, what we've done so far is we've looked at extracorporeal life support and systems that could replace our lungs and kidneys. Today, we're going to look a little bit at the skin as one of the important interfaces of our body, and therefore it's going to get a little bit closer to the body than those systems that we've previously studied. However, in one sense, because all of those systems have blood circulation running through them, they are already very close to the body. In fact, they're designed as if they are within the body, meaning that internally they're in contact with blood with a significant flow rate, meaning that you get a flux of all the materials that are going to reach various regions in the body coming through these systems. So it's actually engineering those systems isn't a small challenge at all. So what we're going to do is just following that same structure. We're talking about this last point here, the sixth point of this week's lectures, which is biomedical devices, implants, and innovations. And then, as I mentioned, we'll get back to disease in the excretory system and also the mechanics of this system later on in our asynchronous and synchronous lectures. So one of the important medical solutions that is going to involve the skin tissue is sutures. And this is a big area in materials science and engineering, is designing and optimizing suture materials. But to understand that a little bit more and to put it in context, let's revisit the anatomy of our skin. So in our skin, we have this layered structure all the way from the dermis and epidermis to those subcutaneous layers underneath. And what happens is that when you have a bruise, when it actually cuts through the skin, it can be different levels of depth. It can be all the way to the bone, okay, and the internal tissue, or it could be very much superficial. So it actually depends on what level of involvement of the tissue you have in the scar or in the wound. And usually repair mechanisms operate in this simple five-step process here. It starts with coagulation and blood clotting, okay, which is the basic immune response. And then you start having epithelialization and the formation of an epithelial layer. This is the equivalent to the endothelial layer that you have in the blood vessel wall. And so you have epithelial cells forming a layer here. And then you have new blood vessels growing in the damaged tissue area. And then you have the secondary effects coming from those fibers and extracellular matrices secreted by these cells, which are mainly collagen. And that basically retains the sort of recovers the properties of the original site and restores it back to the healthy condition of skin. And then ultimately, as this continues on, the skin starts to contract and the tissue damage and the scar starts to disappear or minimize as it goes closer and closer to the previous condition. So really, that's the wound healing cascade in a tiny nutshell. But when you look at suturability and sutures in general, you're looking at repairing this damage and helping to accelerate the repair mechanism. And what these sutures do is they hold together, of course, the wounded region. And that's exactly why it's critical to understand what their role is in the repair process and also what material properties we expect them to have. So as you can see in these images here, they usually dig deep into the epidermis and sometimes a fatty layer. usually doesn't go as deep as the muscle cells, unless if the wound is really that deep. But you can see there are various styles of doing this. Okay, so here, for instance, you can see the simple one, the regular suture that you have, where you're actually going around down through the dermis and epidermis, making a knot and doing this multiple times. Alternatively, you can have this continuous system where you're actually going from one side to the other, then down, back to the other side, and there, it's a loop again, okay? But usually this is the case. You have various sutures as a loop there, and the idea here is that they would gradually degrade in the case of degradable suture materials and get dissociated here. Sometimes you have a stronger connect down here if it's much deeper, if it goes down to the lower layers of the skin, the hypodermis. It actually is then required to go deeper and make a knot that is in the epidermal layer and actually connect it there. And so there are various types of these architectures. So here you can see those same stitches here in a very simple style. You see that what they call horizontal mattress style. And you have sort of diagonal, continuous, so you can go all around. And it can be a superficial one, or it could be buried underneath in the hypodermis and dermis regions, as I showed you for the case of more severe and deep damage. It's interesting that billions of these sutures are used annually, and this means that this is a big market, this is a big industry. And the idea is that this device, this system is there to help the healing process and also take some of that load, some of that excess rupturing hold that's trying to make that wound even wider, sort of resist some of that in order to help tissue to do the repair. And the idea is that these materials can also fail, very much like any other biomedical device, any other material used in those devices. Sutures can also fail, and we'll look at that in a minute. But usually, it's first of all, the knot that you make there is really important. How strong that knot is can actually open. And the knot can actually be the region of failure in your loop here, because it's actually in that region of the suture, you're having material compressed, and so you've got a complex loading in that region, and that makes some areas in that tiny, if we zoom into that tiny knot, some areas of it more prone to rupture and cracking. Okay, the rest of the material as well can fail, depending on, again, what sort of knot you use. But it's interesting that suture doesn't have to be of this style. It can be these strips that we now use. If you want to apply something really fast, if you want to, if you're in a condition where you don't have access to those kits, and if you don't have the experience doing that, this is the best way to do it. And then there are also glues. This is when you have significant bleeding and usually don't have enough time to do this. You apply, there are these special glues, biocompatible biomaterials that actually seal it quickly in order to allow for the material to start healing. So if you look at the a standard pack that comes, the suture material, the thread comes with it, you'll see that it's usually not just the thread, it's sold with the needle included. Okay, so the needle and the thread come in a single package. And there's lots of terms here, as you can see, to describe this combination here. Okay, in terms of of course, manufacture and the name, but then in terms of the diameter of the suture, in terms of the gauge, in other words, the length of that suture thread that you've got there, and the shape of the needle here, what sort of arc radius it has, which they sort of explain in in terms of how what portion of the circle does it consume. So you can back calculate curvature from that, but it tells you it's got 3/8 of a circle in terms of its perimeter. That's their own language, their own literature to talk about this, and they're comfortable with it in the clinical space. And then you've got, of course, the needle size, similar to the thread size. As I mentioned, the curvature of it, the shape of the needle. You've got the material for the needle as well as the material for the suture here. You've got monofilament polypropylene, which we'll learn about what that means in a second, but also the needle material. And the idea here is that the needle is basically connected to the thread and is sold as a single package. And the reason they do this is that the surgeon doesn't need to worry about, in that timely fashion that they're trying to operate everything, they don't need to worry about sort of threading the needle, so needling the thread, or basically wasting time in finding that hole and connecting these two and all of that. So it's typically sold as a single package. And these the thread materials can vary. They could be natural materials, such as silk or catgut. Catgut is, as the name suggests, derived from the intestines of animals, some cases sheep, horse, goats, and it's mostly made out of collagen, so it's very strong fiber that exists in the intestines, in the intestinal walls, and they extract that and dissociate that in the lab or in the manufacturing plant. It can be an absorbable material that's synthetic. Okay, so natural ones, of course, are absorbable, but there are also synthetic absorbable ones, such as PGA, PLA. They could be, in various other formats and material combinations. And then you've got the non-absorbable ones, which you are hoping to then recover or remove afterwards, such as polyester or polypropylene. There's even stainless steel that is used, so thin down here. You've got a composite material there. So there's variety of materials, but the choice is either to stick with an absorbable material that would gradually degrade, or with something that stays there and then you extract it. So it depends on whether what procedure it is you're looking at. If it's dental procedure, if it's something that you don't then want to have another visit, you want it to naturally get absorbed. If it's inside the body, if you're suturing an internal organ, you want it to be absorbable, so you don't need to have to worry about another repeat procedure. If it's external, sometimes you can then remove it. It's easier to go with a non-absorbable one. And in terms of their geometry a little bit more, as you saw, the previous one was showing in the packaging monofilament polypropylene, and that's what it means is that monofilament versus multifilament is when you've got sort of this braided structure where you've got an entangled, intertwined configuration of multiple strands into one single thread. And in the other case, you've got a monofilament, which is a single material. You don't see it here. It's just that single strand that you've got. Usually we're using multifilament because you can have many different benefits from that. One can be the flexibility. They're usually more compliant. They're also much stronger because of their configuration, as you can see here. Enables an added strength, but they're very much, as I mentioned, more flexible. So if you need something more rigid and sturdy, you want to use monofilament. Okay, if you want to hold it without losing that too much, if you want to hold the tissue without losing that distance too much, or if it's a site where you think it will be under tension and stretching, you may want to go with monofilament. But then there are other advantages too with multifilament, including degradation. Okay, so With the multifilament structures, you can actually have materials involved in these that have different degradation rates. And so at different points of time, as one material leaves this complex, the material properties of the complex will change. And so you can design your recovery process around that. So this could be very much engineered with these new materials. And of course, as I mentioned, the needle is an important part of the problem, although it doesn't stay there, but it's there to assist the process. The diameter of it as it cuts through the skin is important. And as I mentioned, they're usually bonded together. Here are a couple of examples of suture materials. And what you see is here the comparison of those materials. They each have a different timeline of degradation. So what happens is, as time passes, the strength of these sort of filaments or the threads is actually gradually going to decrease. And the reason for that is they either degrade and gradually lose their properties, or just under that tension, they gradually soften, absorbing the moisture and also getting sort of again degraded and consumed by the cells around. So this is for these degradable materials that you can see here. And this is showing also the, in terms of time, the change of tensile strength, but also the weight. So as you can see, they're gradually degrading, meaning that you're actually losing weight, but it doesn't happen until degradation hits a point where these materials are consumed. It's usually the coating around these protects them for a while until it starts losing weight and being absorbed to the matrix. In terms of testing these, there has to be extensive testing beforehand. So depending on whether you're testing the knot quality or a certain knot geometry, or whether you're actually testing the thread itself. And this is really important because you've got to see what is the site of failure, okay? In this case, what we're looking at really is maybe we want to figure out the answer to that, okay? Is it actually the thread itself that fails first, or is it going to be the knot? So they're going to stretch these two drawers, and these are custom designed fixtures to allow us to do that. This is a similar process where you can actually have a knot region here or just the single thread, but it's just stretching one line of it rather than the two sides, which may give you a bit more information. Now, this is something we did in my lab to test the hydrogels. This sort of the suture strength is actually a very critical challenge here. Isn't just the suture materials, it's actually the material that you're trying to suture. And in this case, what we were interested in wasn't the human tissue, it was a mimic for human tissue, which is the hydrogel, the biocomposite that we're using. We want to see if it can actually take that torture from the suture thread cutting through the gel. And this is really important because if you're thinking about implanting these gels, you want to suture them to the native tissue, and therefore you want to be able to maintain that strength there without the material itself failing. So a big aspect of this is these threads are very strong, okay? It's rarely the case where the thread fails. It's actually usually the tissue, especially in the case of these regenerative medicine solutions where we're making a tissue mimic in the lab, is that material isn't strong enough and it gives up. And in terms of, again, the absorption rates, typically we're looking at somewhere between, a month to 3 to 4 months, and even 6 months in some cases. So you can design so you can select your suture material to enable various absorption rates. And here you can see you've got 5 to 7 days for the gut materials, okay, a few days, but then you've got synthetic ones and other candidates that using monofilament materials, and they have pretty long duration here. So this, again, the material choice there does influence the performance that you get in terms of degradation and also in terms of their strength. Okay, so this is basically the entire objective space of designing these sutures is to meet a certain absorption rate and the strength. And what you've got here is the breaking strength retention profile, and it tells you that the retention is about 83% after 14 days there. And this varies also significantly between these different threads. So another thing that you'll see is that with these threads, the performance varies through time, as we saw, because it's losing its material, and it's also potentially losing its strength. But in terms of the response, you get a very strong response after a day, and then typically you get a stronger response for natural materials, of course, because they're easier to degrade and dissociate. And here you can see that sort of in terms of the variations in the polymorphs in the thread region. You can see that it peaks at around 1 day and then suddenly starts dropping beyond that afterwards. And we'll talk about what this means in a bit in more context later. But the important thing here is that there's temporal evolution of the suture properties, including the strength and absorption as well. And this usually kicks in at about 1 to 2 days. So after that, it gradually drops and then becomes constant through time until it's fully absorbed. But the problem doesn't, sort of the solution space for the problem doesn't end there. As I mentioned, you also have those adhesive strips, or you have in the case of the glue, a material to coat the wound region. But you also have solutions such as staples. And these skin staples, again, are when you are facing a very large, sort of a very deep tissue scar. It's bleeding significantly, the wound region, and you actually want to quickly apply the suture. And usually these have, again, like staplers, a system to deploy them, punch it into the skin. And of course, aesthetic wise, they're not good, but they're really fast. Okay. And it's when the loading is significant, you actually want to close it, keep it shut there. And some of these actually use shape memory alloys, and we've visited these materials before. They respond to heat, for instance, and if you heat these staples after they've been deployed, they will contract and pull together the two sides of the wound in order to sort of allow the bonding and the repair. And again, in terms of glues, cyanoacrylates aren't the best material, but fibrogens are a little bit better. But still, we don't have an ideal material. They're now looking at collagen pastes to apply there. Collagen is very expensive, of course, if you need it in large volumes, and if you need it purified with the properties to enhance skin regeneration. But the good thing about these glues is they polymerize quickly, and usually it's either a chemical polymerization, so it's in the presence of another fluid, or it's actually using light with a UV cure system, or sometimes even temperature. Okay, but it's usually a chemical reaction there. And what happens is they get stronger and stronger through time, depending on what sort of chemistry you've got there. And you usually get better strength properties in fibrin and fibrinogen, and in terms of their adhesion strength, and also in terms of the strength of the layer that you've applied there. So there's more tendency to move towards those, but they're also more biocompatible and degrade and integrate within the skin a little bit better than cyanoacrylates. And now we're going to talk a little bit about a slightly different topic of skin patches. And nowadays, these have become a lot more innovative and broader in the application space than they were previously. Previously, these patches were usually to sort of seal a tiny opening or a wound in the body on the skin, very similar to plaster. And now they've actually become multifunctional, so they can actually perform various functions on the skin. They could be used to dress a wound, but in conjunction with doing that, they do various other um, functions as well. Here you can see two examples of these. One which, as the looks of it suggest, has some circuitry in it and a conductive material, and we'll see why that is and what it's actually doing there. And this one has sort of a fluidic channel running around it and some colored chambers here. So this one actually, to start from this one, is a sweat sensor. So it's a skin patch that pulls or basically absorbs the sweat from your skin as it runs and circulates through. It's going to come into contact with these four regions, these four chambers, which each have a sort of colorimetric assay, if you will, which changes its color upon the reaction with the ions or the salts in your sweat. So here you've got a lactic acid sensor, you've got a chloride sensor, a pH sensor, and a glucose sensor. And the good thing about that is you can dynamically sense how the secretions from your body are changing, and it will reflect your internal conditions as you're exercising or as you're in the hospital, basically going through a procedure. So these are usually multifunctional, where you can use different chemicals that react with that biological fluid. And in this case, they're using these for sort of readouts. But in general, we have various other sites. For instance, as I mentioned, you've got the lenses, the contact lenses that have now also been equipped with diabetes monitoring systems, where you're actually looking at, again, that meniscus fluid, the biological fluid in the eye, to actually measure that. You actually then use some similar form of a chemical reaction there. You're not really looking at the color change, but you've got a tiny sensor there that's based on a biochemical species, changing its properties, and that potentially changing the resistance of a piece of circuitry, which then communicates itself to the sort of the smartwatch or the other interface that you have via the antenna. So here you've got a similar one, but it's much easier to work with. It's much simpler. It's got an indication, and you can quickly tell what the condition is. So another example here, you can now print using inkjet printers circuitry on top of these plastic sheets. And what you see here is that very thing. So they've actually printed these tiny sensors, which are very much similar to a strain gauge. And what happens is that they can, sense deformation or displacement of the skin, the muscles underneath, but they can also potentially perform ECG, okay, and read some of the electrical signaling underneath if they've been designed as a capacitor. There are various things that you can do here. You can lay down multiple layers of electronic materials in order to produce various architectures, and then this goes on the skin. Here you can see a variety of those materials that they can print using inkjet printers. You can print silver nanowires, and each of these have their own properties and integration within this sort of sandwich structure. What they're doing here in Caltech is they're using these readouts to guide the motion there. So it's rather similar to a haptic system, but here directly reading it from the nerves on your hand. It's actually looking at those electrical signaling processes happening under the skin, and so translating that into a signal which guides them onto, are you now closing your fist, are you opening it, and what the robot should do sort of to mimic that. And these are a couple of examples. So these were a couple of examples of smart devices used in skin patches. And with that, it's now a good time to also talk a little bit about skin patches that are used for regeneration. And a good example here is the Integra skin. And these are a level higher than what you expect from sutures. When you're suturing, you're just trying to hold the tissue and allow it to remodel and regenerate. With these artificial skins, you're looking at also helping it in its regeneration process, speeding that up in this system. So this example is a good example because it actually has a sandwich of silicone and collagen. So you've got collagen and glycosaminoglycans as a porous, sort of macroporous material underneath. And then you've got the silicone coating up there. And what it does, the silicone itself is actually preventing the fluid loss. It's also helping with managing bacterial ingress. And the membrane, so the inside region, which is a 3D mesh, is helping the clotting mechanisms and regeneration and the cell accumulation underneath this layer. So it's helping the epithelialization, in other words. And what happens is once you've done with that and this process is completed, you can then peel it off and remove it. So it's very much like a Band-Aid, but what it does is it actually leaves its internal layer as a matrix to help the remodeling of the epithelial layer. And the silicone there has multiple advantages. First of all, of course, it's going to prevent the fluid loss and bacterial ingress. But secondly, it's the silicone is breathable, so it's going to allow oxygen to pass through, and so the cells are not going to die under that sort of blanket of hypoxia, or basically lack of oxygen. And apart from that, they're not going to be sweating underneath there, although sweating sometimes helps regeneration. That sort of increased metabolic rate, but in general, the breathable aspect helps it feel like it's in contact with air, just like the natural condition, while sort of supporting its growth into a proper functional dermal layer. So this is one example, but then you've got the various other examples too. For example, the BioBrain here is another synthetic wound dressing, and what you can see is it's still using collagen, but it's not a pure collagen layer there. You've got a mesh of nylon, which is really strong, holding it together, and that mesh of nylon is then coated with the collagen peptides. So the peptides are usually from porcine models, and so from pigs, you've got the collagen in their skin, in dermal regions, the epithelial layers underneath those, and that's extracted and then used here, bonded. So these peptides are bonded to the nylon mesh, and that's used as the layer underneath, and then you've got the layer of silicone rubber on top. And this is really the same idea as what you saw here with the Integra skin. And the idea with all of these is to really decrease the length of the patient's stay and the regeneration to be accelerated. It also reduces the pain because of its property to integrate with the skin and hold it all together and coat it, so that you don't feel that exposure to air and to the external environment as much as you do with sutures and other stuff. And so really the idea here is to enhance and accelerate the process of regeneration. Unlike when with the sutures, we are doing that, but we're not actively doing that, meaning that we're not getting involved with the regeneration process as much as we are in these cases. So with this, I think we've had a good sort of comprehensive review of what sort of medical materials and innovations we have with skin as an interface in the excretory system. We'll continue this conversation in a little bit more detail and then conclude and move on to the diseases and material selection for these applications in the other lectures. But for now, let's give ourselves a quick break and we'll move on to those sections after the break. Thank you very much and take care.